My name's William McGowan, work in the University of Hull, and I'm here today to do a talk on mind-body interactions in hypnosis, and specifically the neuroimaging of hypnosis and suggestibility. So we'll look at what effects hypnosis has on the brain, and what relationship there is with the brain with absorption, mental imagery, dissociation, and spontaneous thoughts. So today I'm going to talk about mind-body interactions in hypnosis, and specifically about the neuroimaging of hypnosis and neuroimaging of suggestibility, which I'll come to right at the end of the talk. Looking up the different uses of hypnosis and hypnotherapy, these are the uses I found in the website here. On another section of the website, I found 48 different uses. So it seems to be used extensively in today's society. But what actually is hypnosis? What changes take place in the brain when someone's under hypnosis? Are there any changes, and can we observe those? So we can ask what hypnosis is, and I just took this definition from the Oxford English Dictionary: the induction of a state of consciousness in which a person apparently loses the power of voluntary action and is highly responsive to suggestion or direction. And for the purposes of today's talk, I think it might be a good idea to conceptualize hypnosis like this, in which it's a two-stage process. So there might be the hypnotic induction, in which suggestions for deep relaxation, for example, might go in, pleasant mental imagery, and suggestions to enter hypnosis. And of course, everyone in this room will have their own idea of what should be in that hypnotic induction. And then there might be suggestions which make up the therapy component, or I'll talk about some other suggestions later in the talk as well, and the effects of those. Okay, so researchers in the field of hypnosis, they tend to assess people using scales of suggestibility. And I have an example here of an adaptation of the Carleton University responsiveness to suggestion scale. And there are other ones from Stanford University, from Harvard University, for example. Subjects are given a hypnotic induction, and then they're asked to participate, or they're given these suggestions one by one. So the more of these that someone responds to, the more suggestible they're said to be. And you can see here that in this particular scale, there's a number of ideomotor suggestions, motor challenge suggestions, and then which are deemed the most difficult ones, the perceptual and cognitive ones here. If we look at the distribution of suggestibility in the normal population, we can see that this is on a 12-point scale rather than the 7-point that I just showed. But we can see here that not many people seem to respond to all items on the 12-point scale here. So there's not many people that are really, really hypnotizable. We can also see down here at the bottom of the scale, the low hypnotizable or low suggestible people. There's not many people down at that end of the scale. And we can see that the majority of the people, as you all encounter in your practices, I guess, are moderately suggestible anyway, and can be hypnotized to some extent. So the studies in hypnosis, because neuroimaging tends to be quite expensive to put people in an fMRI scanner, or to put them in a positron emission tomography scanner, for example, we tend to take people that are very highly suggestible, that we know will respond well to the hypnotic suggestions that we give. So these tend to be the people that we study, and the people that you'll read about if you read any of the research articles in this area. As a good comparison group, we tend to recruit people down here at the low end of the scale. On the hypnosis neuroimaging research, it tends to leave out everyone in the middle here. So what happens to the majority of the population, we don't really know, and we don't really study yet. So maybe that'll come at a later stage, but not just yet. Okay, so what kind of features characterize hypnosis? I've listed some of them here. There's many more, but deep relaxation, mental imagery, increased absorption. So focusing on something and ignoring all irrelevant stimuli, feelings of involuntariness, disorientation in time and space, reduced reality monitoring, feelings of dissociation, whether it be dissociation from the external environment or dissociation in some way from the person themselves, and also reduced spontaneous thought. So just to give you a little preview of what I'm going to cover today, I'll focus and I'll show you some of the experiments that show the results of mental imagery, show the results of absorption, and what areas of the brain are related to that, the areas associated with dissociation, and the areas associated with reduced spontaneous thought. So the kind of questions we might ask are, why might people be more suggestible when they're under hypnosis? Why do they respond to more suggestions that are given? And how do we go about testing this? Well, this is one brain imaging technique that we can use, and this is positron emission tomography. And the first set of studies that I'm going to show you, they use this technology, so I'll just explain it quickly. When people lay in the scanner, they're given a small radioactive dose. And this is actually something that's very like glucose, only radioactive glucose. And when the brain works, when your neurons do work, then they metabolize glucose. So we can see here in these images, the brain regions that actually are metabolizing the most glucose. So we can infer from that which brain regions are working most at any one time. And this is a typical PET scan that you might collect. So this is just an axial slice, which is taken through the brain like this. And you can see the front of the brain, the back of the brain. And here it's color-coded as well. So the red and yellow colors, they are associated with a lot of glucose being metabolized. And the blue areas, they mean not very much glucose. So most likely this person was lying in the scanner with their eyes open. And that's why we can see the red areas, the back of their brain, the occipital cortex, their visual regions, they're doing a lot of work here. Okay, so the first study that I want to tell you about is one that was done in 1999 using positron emission tomography, or PET. And it was by Maquette et al. And colleagues. So the idea here is normally with PET or with functional magnetic resonance imaging, the kind of setup of the designs are usually along the lines of a subtractive design. So if you have, in one condition, you get people to do something that recruits processes of their brain A and B, and in another condition, they recruit only B for that, then we can subtract those brain scans and see which brain areas are related to the processing of A. And it's a statistical procedure, but this is what we tend to adopt for PET and fMRI as well. So here we need to choose our comparison groups. So here, this group of researchers, they had someone engage in mental imagery, and it was of imagery of autobiographical memories in the past, which they listened to, and they did that in an alert state. Then they also got people to do that while under hypnosis, so they had to try and relive those memories in their mind, engage in an imagery process, and then actually had a third condition as well, where they had people under hypnosis. They engaged in mental imagery, but they had to try and hallucinate the colors vividly as they did this as well. So when the researchers compared these two groups, or these two conditions, those of hypnosis, they found no significant differences there. So they just merged both of those. So now the comparison we're going to look at is hypnosis versus just being alert in the no hypnosis condition. So with this kind of comparison, we should be able to see what kind of brain areas are used during hypnosis. And this is what the researchers found. So here we can see this is a glass brain. And by looking at these three different types of views of the brain, this one's sagittal view, that's this kind of view down the middle. Here you can see the axial view again, and this one's just the coronal this way through the brain. And we can work out from that then that there's activation down the left-hand side of the brain here, from the posterior to the anterior part. And we also see a lot of metabolisms taking place in the visual areas of the brain. So we might infer that these regions are due to mental imagery processes, and people tend to report that a lot while they're under hypnosis, that they engage in mental imagery. So here we seem to have some brain imaging research to back up those claims. People seem to engage in that in hypnosis. But it could also be the case, because of the choice of task here that these researchers chose, that, and it all being about mental imagery, what if the people in hypnosis are just doing the task better? If they're doing the task better, they're most likely recruiting more visual regions and would expect more metabolism there. So we might not be getting exactly to the heart of what hypnosis might be with this analysis. We might have to look at other types of tasks then. And specifically, if we looked at tasks that don't require mental imagery at all, but we hypnotize people and see visual regions, then we might expect that, okay, these visual regions are associated with a hypnotic state. So we need to look at some other types of tasks. I just want to digress slightly for a minute and talk about hypnosis and pain perception, and just show some results of how hypnosis can be used as a tool to try and manipulate different aspects of pain. So if someone feels pain, there's not just one area of their brain that's activated, but it tends to be a number of brain regions that make up a pain matrix. So two of the areas that tend to be activated are your somatosensory cortex, which seems to be related to the intensity of the pain they feel. And there's also a different component, which is the affective component of pain. So how disturbing that pain is, or how unpleasant the person feels that pain might be. And the brain region associated with that is the anterior cingulate cortex. And I've just got a diagram, front of the brain, back of the brain, and you can see the cingulate runs right down the brain here, but the anterior part here, that's what's associated with the pain response. So these researchers, they thought that maybe they would try and manipulate the pain network independently, different parts of it independently with hypnosis, and see if that was possible. So they gave suggestions to the people in this particular experiment. They had them engage in a painful task, which was put their hand in hot water, and it was water that was 47 degrees, so they report feeling pain with this temperature. And they gave suggestions to the participants that they would either feel more pain, so higher pain intensity, or less pain intensity, and only manipulated that with suggestion. So the actual temperature of the water, it remained exactly the same. So what we see here is when they suggested a higher intensity of pain, an S1, which is the somatosensory region, we see lots of metabolism. When they suggest less intensity of the pain, we see less metabolism. And if we do that kind of subtraction I talked about earlier, one from the other, we can see that there's a significant difference here in the amount of metabolism that took place in their brains. So here we can see that just by suggesting more or less pain, we seem to see manipulation or modulation in the somatosensory cortex. They also looked in the anterior cingulate, and they didn't give any suggestions to try and manipulate how pleasant or unpleasant the pain was. And here we see the same level of activation in this brain region. So they seem to succeed in only manipulating part of the pain matrix. They also set out in a different set of experiments to do it the reverse way. So they thought, can we manipulate how unpleasant people consider the pain to be, but leave the intensity of the pain the same? And you can see here, by just giving suggestions of how unpleasant the pain should be, we see what are no significant differences here in the somatosensory regions, but we see lots of metabolism here when high unpleasantness is suggested, and a lot less metabolism when low unpleasantness is suggested. So here, they seem to back up those clinical reports that have come up in the past about people maybe feeling, they say they report feeling pain, but the pain doesn't bother them. So here they can actually show, just by using hypnotic suggestion, that parts of the pain network seem to work independently of each other. So these experiments, they can also, when analysed a different way, they can tell us a bit more about the hypnotic state and tell us what changes take place when people are hypnotised. So they had a hypnosis condition and they had a restful awake condition where people aren't in hypnosis. And the first thing they did was they had the painful conditions. They also had warm conditions where the water wasn't painful, just as a baseline condition. And here they put both of these together, and they put both of these together, and they thought, okay, we'll just see what happens in hypnosis versus rest. And this is what they found. So when people are hypnotized, they found that there was activation or more metabolism in the visual cortices at the back. So it seems people did engage spontaneously in mental imagery, and asked people afterwards, what were you doing during this task? And they said, oh, when I entered hypnosis, I did engage in mental imagery. So we see that coming out in the brain scans here, and we see that this seems to be one characteristic of the hypnotic state. There's also activation in the anterior cingulate. So this region also seems to be important and involved in some way in hypnosis. So another thing we can do is, and the researchers, they compared only the painful conditions, hypnosis versus rest, and then separately they compared only the warm conditions, hypnosis versus rest. And this is what they found. So here we can see in the neutral condition, we see lots of activation in the visual cortices. So here people seem to be using lots of mental imagery when the water's not hot and when they enter hypnosis. Here in the painful task, we don't see that. And the participants, again, they were asked what's happening here, and they said the painful condition, it seemed to interfere in some way with their hypnotic state, and pain's attention grabbing. So they said that it seemed to stop them engaging in this mental imagery process. So the hypnotic state, if I can call it that, so far what we've seen in those studies, comparing hypnosis to the alert conditions, there seems to be activation in the visual cortex and this anterior cingulate region. The anterior cingulate, it's a very heterogeneous region, and it's associated with emotion, it's associated with attention, and we'll see probably a bigger relationship to attention later in the next slide. Using the warm neutral condition, there seemed to be lots of visual areas which were activated, and also the anterior cingulate. And in the painful condition, pain was reported to interfere with the hypnotic state, and activation in the visual areas, it seemed to be reduced substantially. So something that this kind of flags up is the task we use to study hypnosis seems to be really important here. If we didn't have that neutral condition, if we just looked at the painful conditions only and looked at someone not in hypnosis and in hypnosis, we wouldn't have seen any of the visual activation. So we would have just said, okay, the hypnotic state, it's all about the anterior cingulate, and that's it. So here the task seems to be vitally important. Whatever task we choose, it might have a differential effect on what we'd regard as hypnosis in the pattern of results. The researchers also analysed their results in a different way, and this is the statistically controlled for the conditions, so whether it was painful, neutral, hopefully that doesn't matter anymore. They tried to remove any effects of that. And what they got their participants to do was they got them to rate how much absorption they felt while engaging in these tasks. So how focused they were on the task and how much they were ignoring what was going on around them. And they saw that with greater absorption, there seemed to be more activity going on in the anterior cingulate cortex. So there seems to be this relationship here, and further support that the anterior cingulate's an important brain region for hypnosis. So the anterior cingulate in hypnosis, we've seen that in the mental imagery study, the very first one, that I showed you. It wasn't displayed, but they also found activity in this anterior cingulate, this attentional brain region. In the studies which investigated pain, we found greater activity in that in the hypnotic conditions compared to non-hypnotic. And we also find that increased absorption seems to be related to this brain region. So it seems to be a really important brain structure for hypnosis, and we'll see a little bit more about it at the end of the talk. We've also seen some of the characteristics of hypnosis. I said we'd look at some of what the neural correlates are in the brain. We've seen that we can see visual areas associated with the mental imagery that are activated when people are under hypnosis. We've also seen increased activity in that anterior cingulate region when they're reporting higher levels of absorption. So let's move on to a couple of the other characteristics. We can also use magnetic resonance imaging as a neuroimaging technique. So there's a couple of different types of brain scans we can acquire here using MRI. And the first example is structural magnetic resonance imaging. And here we can see it's a very high definition image. This is again just a cut through the brain in the axial slice. And we can see the grey matter, the neurons that actually do something. We can see the white matter distinction here. So these are the fibers that connect the different brain regions. And we can see functional MRI, which seems to be a lot lower resolution. And the pros and cons of each of these is the structural MRI. It takes about 5 minutes, 7 minutes to acquire a full 3D brain image. Whereas with functional MRI, it only takes about 2 seconds to acquire the whole brain image. So functional MRI, it gives us the opportunity to actually look at blood flow changes in the brain and to see what brain regions are being used at any one time. So this is a slightly different type of analysis, and it's functional connectivity. So it looks at correlations between brain regions, kind of how much the brain regions are talking to each other. And these researchers, this was just published this year, they had 3 conditions. They had just the waking state when someone's alert, and these are the correlations they find in what's called the extrinsic network, or this external network. So this is associated with when people are attending to what's going on around them. And when you or I are just sitting in the audience, for example, there's lots of different brain networks that are spontaneously activating, and they're actually competing. So when one's active, the other's not active. And this is one of the networks that they've isolated and highlighted here. And this is the network that's for attending to things happen in your environment. When you're attending to visual scenes, auditory scenes, anything touching your hand, for example, you see activation and connectivity, an increase in connectivity in these networks. They also study people not in hypnosis when they were engaging in an autobiographical memory task, and also people in hypnosis engaging in the same type of task. And we can see here, when not in hypnosis, we see there seems to be a decrease in connectivity and a decrease in attending the external environment when people are doing this task, as you might expect, because autobiographical memory and the recall, it's a pretty much internal task. You don't have to pay attention to what's going on around you. So we see this slight decrease in connectivity. But here we see when people engage in hypnosis, we see that people seem to stop attending to the outside world, and there seems to be this big decrease in connectivity in these brain regions. So this in some way looks to me like it could fit with dissociation and this kind of mental separation from the environment that people report. And in fact, in this study, the people went under hypnosis, they did report higher levels of dissociation and also higher levels of absorption. Okay, so with all the types of tasks I've talked about so far, they tend to have been hypnosis with some kind of suggestion to try and increase the intensity of pain, decrease the intensity of pain, retrieve autobiographical memories, for example. So we need to think about it with these types of tasks. Are we studying hypnosis per se, or are we studying hypnosis and suggestion combined? So we thought about this, and this is where our studies started to come into this. And we thought about it would be great if we could study hypnosis but without these suggestions. And we thought we'd try and set it up in a way that that might be possible. So our task, and what we did in an fMRI scanner, is we got people to engage in a colour hallucination task. So people were doing a task, but the parts that we actually looked at were during rest. So we set up the task that they'd have rest periods. They'd look at a grey Mondrian image, and this is based on a study that you might have heard of by Coslin in a colour hallucination task. And I'll show you the results of that study a bit later. But people looked at a grey image, and then the next part, while they still were in the scanner, and we were collecting brain images, looked at the grey image, but they had to try and hallucinate colour. So they'd had to try and see colour. Then they'd rest a bit more, then they'd look at a colour image, and then they'd have to drain colour and try and see only grey in that image. So we have a kind of positive hallucination and a negative hallucination here. And in between all of these, we collected these rest scans as well. And we did this while people were in hypnosis. We did this while people were out of hypnosis. And we also collected it in people that were highly suggestible and we should be able to hypnotize. And we also did it on people that are very low suggestibles that reported that they couldn't be hypnotized. Okay, so we thought it would be great not to look at these these sets of scans at all initially, but let's look at what happens just in the rest periods. So comparing the resting state here, this seems to be regardless of suggestion, because all the participants, they're given exactly the same suggestion when they're in hypnosis or out of hypnosis. So the effect shouldn't be due to that. Task demands are really low. It's resting state. The people aren't engaging in any colour hallucination task. In fact, at that point, they shouldn't be engaging in anything. We haven't told them to do anything apart from rest. And demand characteristics are minimised. So if people played along and said, oh yeah, I'm hallucinating colour, in the rest periods, they're not expected to do anything. So they shouldn't try and conform in any way to what we would expect them to do. So what do we find when we compare hypnotic induction and rest and no induction and rest? We see before I've showed you activation on the different brain maps. Here we can see deactivation, so this is lower activity, and it's lower activity under hypnosis in the high suggestible participants. So we see lower activity in the medial frontal gyri and in the anterior cingulate. So they seem to be suppressing the activity in these brain regions. And we'll come on to what these brain regions might do in a minute. So it was important to us to study the low suggestible people as well, because if we say that these effects are due to hypnosis, then we shouldn't see the same effects in low suggestible people. Otherwise, it would be an effect of the induction that both can respond to. So we looked at the low suggestible participants, and we saw that there was lowered activity in a brain region called the thalamus. So this tends to be a brain region that's associated with wakefulness and alertness. And we think that maybe the low suggestible people, they were responding to part of the hypnotic induction. When we say to relax, maybe that's what's causing the activity in their thalamus to go down. So they're actually listening to that part of the induction and engaging in that instruction. We found that there were no significant differences in the frontal lobes here that we found in the high suggestible people. So there seems to be something different going on in the people that report high levels on deep hypnosis compared to these people that report being unable to be hypnotized. And this is just a subtraction which shows there was significantly lower activation in this medial frontal area in the high suggestible people when under hypnosis. So what's actually happening in the hypnotic induction? It appears to affect the high suggestible people differently to the low suggestible people. On the focused deactivation, it seems to be in these medial frontal regions. So I just want to introduce you to the default mode network. So I mentioned when you just sit there and you rest, if you daydream or let your mind wander, for example, there's a certain brain network that's activated called which has been termed the default mode network. So this is a different one to that kind of external network that I mentioned before. On lots and lots of neuroimaging studies, not in hypnosis, but in all kinds of things, they set up these kind of subtractive designs where they'd get people to do a task, they'd collect them just resting, they'd subtract those and find out what brain regions did they use during a task. Well, that went on for about 10 years of looking at what brain regions do people use during different tasks, when some researchers thought, I wonder what happens with the opposite comparison. So what actually happens when people are at rest? What's their brain doing the majority of the time? So when you're thinking about the future, thinking about the past, using your autobiographical memory, for example, you see activation in this network, and this is what the researchers showed. So there's medial frontal regions here, and there's also a medial parietal region. Okay, and not all of these brain regions do the same thing, but we'll come on to that in just a second. So here, this is the study that I just showed you. This has been replicated lots and lots of times, this very robust finding that there's activity in these brain regions when people are at rest. And we can see this is just a meta-analysis and lots of studies, and they show the same kind of pattern here as the earliest studies. And what we can see in our brain imaging study on hypnosis here is they seem to deactivate, and there seems to be quite a good correspondence to the medial frontal areas that are used when people are daydreaming. And the medial frontal regions in particular, they tend to be used for self-referential thought. So when people are thinking about themselves in some way, and when they're having this spontaneous mental activity, they're activating these medial frontal regions. And it seems that when in hypnosis, people are shutting down those brain regions, so they're having less spontaneous thought, and especially what seems to be less spontaneous self-referential thought. And we suggested in the paper that was published that this could correspond with a kind of preparatory state where people are reducing that activity so that they can respond better to suggestions or respond better in certain tasks. Okay. I think I'm just going to skip that slide. I'll go through it just very quickly. And just the implications for that particular finding in future hypnosis studies. When people do a task, you see that default mode activity, it decreases. And you see when people are hypnotized, that default mode activity decreases. So if you're to get people to do a task while under hypnosis, you're very unlikely to see any difference comparing people under a task when they're not in hypnosis, because both of those things reduce that brain activity. So it's just something to bear in mind when we set up future research studies. Looking at differences between high and low suggestible people, and thinking about neuroimaging of suggestibility here. In hypnotherapy, you might get people to modulate pain or reduce anxiety, for example. I talked about our task of getting them to hallucinate colour, this kind of perceptual change. There's also tasks in getting people to have cognitive benefits, like improve their attentional focus. But I just want to talk about our task, this colour hallucination task. So I mentioned before it was based on the study by Coslin in 2000. And Coslin got people to look at this grey Mondrian image, and he got them to try and hallucinate colour as well. So this is the activity in a brain region. It's the left fusiform region, which is a brain region associated with colour processing. And what Coslin found was, when not in hypnosis, people, high suggestible people, when looking at grey, they had this level activity in that brain region. When they tried to hallucinate colour, and they said that they were hallucinating colour, they didn't say they were hallucinating colour at this stage, not in hypnosis. We see no change there. So this is just looking at grey. This is trying to hallucinate colour, not in hypnosis. This is during hypnosis. We see that looking at grey, and we see this boost in activity in this colour processing brain region when people report hallucinating colour. So here we see that as well as people saying that they're hallucinating colour, we actually see that the colour regions of their brain are being activated. So we have strong support for whatever they're saying is actually true as well. So this was an important study back in 2000. There was one problem with Coslin's, well, a couple of problems, but one that I want to draw your attention to with Coslin's study, and that's that between the no hypnosis and the hypnosis conditions, Coslin didn't seem to keep the suggestion the same that was given to the participants. So again, we have this kind of worry, are the changes we see due to hypnosis and suggestion, or only hypnosis? So the idea here is, Coslin said, when under hypnosis you should hallucinate colour, and said when not under hypnosis, try and remember the colour from before and try and image that in some way. So here the suggestions weren't quite the same. So in our study we had it set up that the people not in hypnosis and in hypnosis were given exactly the same suggestion. So they were told that they should try and hallucinate colour, actually try and see colour when looking at this grey image. And this has just been accepted for publication, so it'll be coming out soon. And consciousness and cognition. Okay, so we had it set up, this kind of 2x2 design where we had hypnosis, we had no hypnosis, and we had high suggestible people, and we had low suggestible people. So we can look at all combinations. And we found in the high suggestible people, this just a cut through the brain, mentioned this colour processing region. I've just highlighted it with the crosshairs here, and we see activity in that brain region. So when people report hallucinating colour, they seem to be activating this brain region. So essentially we've replicated what Coslin did before. And we can see here, we actually collected reports about the intensity of the colour experience that people reported as well. So here we can see that this is the high suggestible people. This is them looking at the grey. And this is a colour scale from vivid colour to grey, not much colour at all. And we see that this is no hypnosis, this is hypnosis. We see a boost. So hypnosis, and it's a significant boost. People report seeing significantly more colour when under hypnosis trying to do this task. If we look at the low suggestible people that can't be hypnotised, they don't report being able to do this task at all, even when we give them the hypnotic suggestion or the hypnotic induction. So they just say that they don't see much colour even when they try. So here we can also, on that last slide, we can also see here that when not in hypnosis, so without any hypnosis at all, people seem to, in the high suggestible group, they seem to report seeing quite a lot of colour here anyway. So this is with no hypnosis. So we looked at the brain images associated with that, and we saw that there was activity in their fusiform region. So regardless of hypnosis, they were managing to do the task, but hypnosis seemed to provide this extra boost. And when we looked at the low suggestible people, there was no activity in their colour regions. So here this suggests to us in some way that the people that can be hypnotised, they seem to be in some way special. There seems to be something different about them that allows them to do this task, whether with hypnosis or without hypnosis. But hypnosis does provide that extra benefit. So we compared the hypnosis scans to the no hypnosis scans, and we did see modulation in the visual cortex. So with hypnosis, we saw greater activation in the visual cortices than without hypnosis. So focusing, or just a mini summary first on hallucinating colour. The high suggestible people, they can perform this task whether in hypnosis or out of hypnosis. The low suggestible people, they couldn't do the task at all. And the results suggest to us, anyway, in some way that the high suggestible group's in some way special. And that's prompted us to try and investigate this further and to have a look at what could be different about their brains that allow them to do these extra tasks or allow them to be hypnotized deeply. And we did see hypnosis seem to provide a boost to their colour adding ability, however. So just focusing on differences in suggestibility, so looking at the differences between high suggestible people and low suggestible people. There's only one study in this looking at anatomical differences in the brain that's been carried out by Horton and colleagues. And before I mentioned the anterior cingulate, there's this region below the anterior cingulate, which I've highlighted here, which is called the corpus callosum. And this is the network of fibers in your brain that connect both hemispheres. So it should be associated a lot to attention. So these researchers, their hypothesis was that at some point, this corpus callosum region should be different between high and low suggestible people. So you can see on a MRI scan, and this is the structural brain scan, the one that takes a long time to acquire, we can see the cut just through the head like this, and we can see their corpus callosum. It's very easy to see, and what these researchers did is they drew around that and they measured the area of it on multiple slices. So here they divided it into different sections, and what they found was this region that's marked 1 in the diagram here, the rostrum, which is just the most anterior part of the corpus callosum. It seemed to be bigger in high hypnotizable people, so they seemed to find some underlying anatomical difference between these two groups as well. So we've decided that we'll use this other approach called voxel-based morphometry, and it's an automated technique. So we just take the person's structural brain scan, high resolution, and we can actually just tease out the grey matter using this automated technique. And then we can compare different groups of people, high suggestible versus low suggestible, for example, and see if there's any regions in their brain that seem to be significantly larger or smaller in size, for example. So we've done that, and but we've only done it on a very small sample of people. So I have just the pilot results of that to show you today, but these aren't published, so I'm not sure how much you should actually take away on this today, but I thought I'd present them anyway. So we'll acquire more people before trying to take this to publication. But what we do see is the high suggestible people, they seem to have a larger anterior cingulate region in this part than the low suggestible people, which might be associated with greater attentional processing abilities, which would fit well with the study by Horton there on the corpus callosum. And we also see that there seems to be greater volume in visual regions, which might account for mental imagery abilities in high suggestible people. Okay, so just the conclusions then. Hypnotic induction in high suggestible people, as they're doing a particular task anyway, it seems to activate visual regions, which seems to be associated with mental imagery, and also the anterior cingulate cortex, this region associated with attention. We saw that absorption seems to be related to this attentional brain region, the anterior cingulate, and we also saw a decrease in the connectivity analysis, that external network that appears to be associated with dissociation and retraction from the external environment. In the absence of any task, hypnotic induction, it seemed to suppress activity in the medial frontal regions, which might be associated with self-referential thought or just spontaneous thought. and it might reflect some preparatory state. And we saw also that that effect's not recreated in low suggestible people. The conclusions in suggestibility. When participants reported changes in perception, the brain imaging data, it seems to support those claims. And we saw that in the color hallucination study that I showed. We also saw it in the pain studies. We see modulation, for example, in their somatosensory cortex. So when they report these changes, we see that their brain, it reflects what they report. We see that the high suggestible people in our task, the color hallucination task, they were able to do it either with or without the hypnotic induction, but the hypnotic induction seemed to benefit them to a small but a significant degree. And final conclusions are that differences seem to exist in the brains of high and low suggestible people, and this is something that we're interested in investigating more. We saw that one published study on increased size of the corpus callosum, that white matter fiber tract in the brain. And we also saw in this preliminary pilot data that there seems to be increased size in this attentional area, in the anterior cingulate, and also in the visual regions. So I just want to mention the collaborators as well, Professor Irving Kirsch, who you heard about earlier today in the talk. He's written the book on the Emperor's New Drugs. He also does a lot of investigation into placebo research as well as hypnosis. His wife, Juliana Mazzoni, is very interested in this field. Another person that works in the neuroimaging area, Annalena Veneri, and these are 3 research assistants that have also worked on the projects that I mentioned. Thanks to the organisers for inviting me today, and also thanks for your attention.